This procedure uses a three-dimensional cell culture system to mimic in vivo cell kinetics. Begin by incubating cells with collagen-coated beads. Then load the cells and beads into a rotating cell culture system and propagate 3D cell aggregates in a low-shear, low-turbulence environment. Finally, harvest the cells for use in downstream applications. Compared to gene expression and cellular differentiation of cells grown in a typical monolayer, the resulting 3D cultures demonstrate cell kinetics and behavior that closely resemble that seen in vivo.
We have used the RCCS to engineer biologically meaningful 3D models of various human epithelial tissues. Dr. Jessica Warner, a former grad student, and my collaborator Dr. Cindy Morris's lab will now demonstrate this procedure with EVT cells.
In a 50 milliliter conical tube, autoclave 250 milligrams of microcarrier Cytodex-3 beads in 12 milliliters DPBS. Allow the prepared beads to swell and cool to room temperature. Then, under sterile conditions, bring the total volume to 12.5 milliliters using DPBS. Now, harvest an 80% confluent culture of Extravillous Trophoblast cells using trypsin digestion. Gently mix the prepared Cytodex-3 beads, and using a wide-tip 10 milliliter serological pipette, transfer 2.5 milliliters to a 15 milliliter conical tube. After the Cytodex-3 beads have settled to the bottom of the tube, pipette off the top layer of DPBS without disturbing the Cytodex-3 beads. Mix the EV